Hello? There we go. Now it works. Good afternoon. Before we begin, are there any announcements from students or faculty? Yeah. Did everyone get that poster drafts by the end of the day to Professor Merritt? Great. All right. So it's my pleasure to introduce Dr. Katrina Armstrong to Ocean Studies this afternoon for our seminar. Dr. Armstrong is a postdoctoral researcher managing the University of Maine's Marine-Aligned Research, Innovation, and Nationally Recognized Education Initiative, otherwise known as UMaine Marine. And UMaine Marine is a unique Maine-based initiative that brings together university, industry, government, and community collaborators who, through integrated and innovative transdisciplinary marine research, education, and outreach, are dedicated to the enhancement of the social and economic well-being in Maine and beyond. Katrina's research interests revolve around the conservation and management of marine organisms and ecosystems, characterized by a transdisciplinary approach that integrates knowledge from genetics, ecology, and local communities. Katrina earned her Bachelor of Science in Marine Sciences and Master's of Science in Biological Sciences from the University of South Carolina, where she studied the population dynamics and life history of the Kemp's Ridley sea turtle and the bluefin tuna in the Gulf of Mexico. And then she earned her PhD from Stanford University, where she identified naturally heat-tolerant corals and their associated symbionts for use in conservation and restoration, and examined the stability that thermal tolerance, and then emphasized the importance of centering stewardship and restoration efforts. So thank you for coming all the way to Castine, and I'm really excited for this seminar.
Thank you. Thanks for having me. It's great to be here today. My first time in Castine and at Maine Maritime Academy, so I was excited to see the space. Okay, so like she said, I'm Katrina. It's great to meet you all. I will share a little bit about my doctoral dissertation work titled The Mosaic Reef, Leveraging Genetic Differences and Local Heat Resilience for Stewardship-Based Coral Conservation. But first, thought I would start just a little bit with my background, which you all heard about already a little bit. But so I grew up in Cincinnati, Ohio, which is far from an ocean. But my mom used to take me to the library when I was little to rent books and documentaries, et cetera. And one day I came home with a cuttlefish documentary and absolutely fell in love with the incredible creatures that they are in the ocean that they live in. And then sometime in like 4th or 5th grade, I did a science project on coral reefs. And from then on was like, I'm gonna be a marine scientist, and here we are. So after high school, I went to University of South Carolina, where I studied Kemp's Ridley sea turtles in the Gulf of Mexico, working in a conservation genetics lab. And in that same lab where when I got my master's, I worked on these bluefin tuna in the Gulf of Mexico, using fish eggs as a tool to more accurately identify spawning location and the number of females spawning. Then I went to Stanford for my PhD at Hopkins Marine Station, where I took an interdisciplinary approach to study coral bleaching and restoration. And that's the work that I'll share a bit more with you today. And now I'm a postdoc at UMaine, managing the UMaine Marine Initiative. And UMaine Marine's mission is to promote interdisciplinary research, outreach, education, and collaboration across industries within Maine and beyond. And right now we're composed of 9 units at UMaine officially, but the UMaine Marine community really extends far beyond these units. And we're working on growing that reach. So if you scan the QR code, you can sign up for our newsletter, which will kind of keep you in touch with what we have going on. There are lots of events and opportunities for people, not just at UMaine, but everywhere. Okay, so before I dive into my work, I want to take a moment to acknowledge the land on which this research was conducted. So first, Hopkins Marine Station's land acknowledgement states, we acknowledge that Hopkins Marine Station sits on the ancestral land of the Ohlone Rumsen tribe and give thanks for the opportunity to live, work, and learn on their traditional homeland. We pay our respects to all Ohlone people, past and present. And the bulk of my work takes place in the archipelago of Palau, and I would like to acknowledge the Palauan people who have stewarded the lands and waters in which this work took place for thousands of years. I honor their deep connection to Palau and recognize the ongoing contributions of their knowledge and practices to the management and conservation of Palau's natural resources. I'm incredibly grateful to have had the opportunity to work in such a beautiful and biodiverse place. Okay, so the overarching goal of the work I will share today is to understand how variation in individuals and systems can inform and improve resilient and equitable coral reef restoration outcomes. But first, what is a coral? So corals are often confused for being plants or rocks, but they're actually colonial animals in the phylum Cnidaria related to jellyfish and sea anemones. And each coral colony is made up of thousands of mostly genetically identical individuals, which are known as polyps. And the colony grows by budding new polyps through asexual clonal reproduction, but corals also reproduce sexually by expelling egg and sperm via spawning or brooding. And this sexual reproduction is how reefs are formed, colonies spread across different reefs. So what are coral reefs? Coral reefs are large underwater structures composed of many coral colonies. The polyps in reef building, or also known as hermatypic corals, extract calcium carbonate from seawater to create a hard, durable exoskeleton that protects their soft bodies. And the polyps live on the exoskeletons of their ancestors. So they add their own exoskeleton to the existing structure. And as the centuries pass, the coral reef gradually grows one tiny exoskeleton at a time until they become these massive features of the marine environment. So I have a little video put together by one of my dissertation advisors, Steve Palumbi, in collaboration with HHMI. And this is going to introduce the concept of coral bleaching, explain a little bit more about the coral polyp, et cetera.
Corals are small animals, but they build structures that you can actually see from space. As we zoom in, we see the reefs where I work, on a little island called Ofu in American Samoa. The ridge there, the edge, is the coral reef. And as we zoom in, we see what those reefs are. They're made of individual coral colonies, like we see now. That coral then is actually made up of a whole series of small polyps, they're called. This is a colonial organism. And the polyps themselves have all the structures that an animal needs. It has a mouth, it has tentacles, it has gonads, it can live, it can reproduce, it can grow. They're in a colony of genetically identical polyps. Now, the color on these tentacles, like I said, is not the color of the coral itself. It's the color of the symbiont. And as the focus racks in and out a little bit here, then what we see is that we can just see the little globules of the symbiont. Well, let's take a closer look. We'll go into a tentacle and see those. These cells, the symbionts, are not just floating around. They're actually inside the coral cells. Corals are simple. They just have two cell layers, an epidermis and a gastrodermis inside. The symbionts are inside the gastrodermis, and you can see it there. This is a life form called a dinoflagellate. It has chloroplasts because it's photosynthetic, but it has very odd-shaped chloroplasts, like these yellow structures here. We're going to zoom into the chloroplast itself because that's where the damage happens during bleaching. What do chloroplasts have in them? They have membranes called thylakoid membranes. Those membranes hold the proteins called the photosystems that then capture light energy and turn it into chemical energy. It's the molecules that turn all of the sunlight that we get on the planet into the food that we eat. The rain of photons down here hits these photosystems and they gather them up. If the temperature goes up and if the light goes up, then they freak out. There's too much energy, the photosystems break, and they no longer can function the way they do. But the rain of photon keeps going. The energy is still there, and as a consequence, that energy is now turned into reactive oxygen molecules. Those are damaging to cells, so it damages the inside of the symbiont, it damages the inside of the coral cell, and they spit the symbiont out. That spitting of the symbiont out by one coral cell isn't bad, but if the entire colony does it, then that's coral bleaching. What you can see here is simulated of the spitting out of these symbionts and the gradual whitening of this particular part of this particular coral colony. Well, when that happens across an entire colony, then the coral turns from its normal tan color into a white color. What difference does that make? The symbiont provides 75 to 80% of the energy the coral needs to survive, and without that energy, it can't make a skeleton and it can't live very long. So as a consequence, a lot of the corals that bleach eventually die.
So that's one of my favorite little videos to show. I think it does a really great job of explaining and visualizing what are coral and what is coral bleaching. Oh, still going on in the background, I think. So now that we know what a coral is, why do we care about them? Well, coral reefs are critically important in shaping ecosystems that provide essential resources to a multitude of marine organisms and also humans. So coral reefs are a hotspot of biodiversity, and this biodiversity creates an oasis of high productivity in environments that would otherwise have very little productivity. They cover less than 1% of the Earth's surface, yet support more than a quarter of all known marine life. They also offer habitat for the millions of species that inhabit them. This habitat is especially important for juvenile fish and invertebrates to protect themselves from predators. And importantly, reefs are not only offer food for the millions of species that inhabit or directly associate with them, but they are a major food source for many coastal communities, and many small island communities rely on reef fish as their primary protein source. So coral reefs play a vital role in the world's economy through the income and resources they provide via tourism and fishing. Tourism alone generates billions of dollars annually for countries associated with coral reefs. Large reefs reduce wave action, protecting coastlines from erosion, storm damage, and flooding. And as you can see in this image, the water is calm by the coast because the reefs are dissipating the wave energy by causing the waves to break. I'd like to just reinforce how important reefs are for humans. So coastal communities of many countries rely heavily on coral reef ecosystems for sustenance, livelihood, protection against the waves, and coastal erosion. One study surveyed individuals in 3 coastal communities in Papua New Guinea to see which ecosystem services they deemed important and why. They broke the services into terrestrial, cultural, and marine. And they found that reefs were valued in both the marine category for fisheries, habitat, coastal protection, and materials, but they also found that they were valued culturally for knowledge, cultural traditions, and the ability to pass healthy reefs on to future generations. So despite their importance, reefs are threatened by human impacts made worse by climate change, such as ocean warming, ocean acidification, pollution, overfishing, sea level rise, and increased storm intensity. These threats are jeopardizing the adaptive capacity of coral populations, so their ability to adapt to these different threats to survive in the future. And many reefs around the world are already experiencing significant coral mortality. Coral bleaching itself is one of the greatest threats to the survival of coral reefs. Bleaching events used to be rare, and now they have become more and more common and severe since the first observed major mass bleaching event in 1997. To combat bleaching and reef degradation, scientists and managers are exploring a range of restoration strategies, including marine protected areas like the conservation areas seen here in Palau, and more active strategies like the propagation of heat-resistant colonies and assisted migration, where you can move healthy corals from one reef to restore a nearby degraded reef. These strategies are still really new, though, especially active restoration strategies, and there are a lot of unknowns surrounding the implementation of these efforts. For instance, identifying heat-tolerant colonies can be expensive and difficult to scale, and we don't currently know if colonies will maintain their heat tolerance when they're moved for restoration. Managers will also need to think about the potential implications of introducing new genes to environments when they move colonies for restoration, considering both the coral host genetics as well as the symbionts genetics. Finally, to make impactful changes in the, make impactful changes in the conservation of coral reefs, we need to figure out how to scale restoration efforts globally and center the needs of reef-dependent communities. So this work aims to provide solutions to these barriers by investigating genetic differences and bleaching resistance of coral and their symbionts to inform stewardship-centered coral reef conservation and present methods that can be affordably and quickly applied in communities across the globe. Specifically, this research focuses on establishing visual bleaching scores as a reliable and accessible means of assessing bleaching phenotype, which I'll go into more, determining the durability of thermal tolerance in coral when moved to new environments, exploring the fine-scale geographic variation and symbiont switching of cladocopium symbionts, and presenting recommendations for stewardship-based reef restoration. So all of my research takes place in the beautiful small island nation of Palau. That little video that you were watching before of the reef fish was taken there. And Palau is located north of Australia and east of the Philippines. The waters of Palau contain an assortment of different reef types, meaning an abundance of sites and species to study. And the Palumbi lab, which is the lab that I did my dissertation in, has 39 reef sites along the Palauan archipelago that we have regularly sampled over the last now 6 years, which are shown here and colored by region. Then we lost some of the colors. So as part of the Palumbi lab's work, Brendan Cornwell led a study, let's see if this will play, there we go, to identify reefs in Palau that harbor heat-tolerant corals. So this study found that different colonies had different levels of heat tolerance, but bleaching-resistant corals were found all across the archipelago in almost every site that we surveyed, so that's good news. Throughout this talk, you're going to hear me refer to heat stress tests or acute heat stress tests, and this is a picture of what those look like for us. We assess a sample's response to heat stress by putting them in these adapted coolers with heaters and chillers and pumps, et cetera, lights. And then we experimentally heat stress them in just little small pieces of coral, less than the size of my thumb, in a time period that mimics the heat stress that they would naturally experience during a tidal cycle. So for us, that looked like about 3 hours, and we did this over 2 days. Okay, so measuring coral bleaching is a really important part of assessing and understanding the health of coral reefs and identifying thermally tolerant individuals for conservation and restoration. However, most of the methods commonly used to assess bleaching today are challenging to scale globally, and the first part of my research addresses this barrier by presenting an accessible means of measuring bleaching resistance that's cost and time effective and can be scaled easily. And here are some folks who helped out on that project. So measuring bleaching phenotype, which is the observed physical response of a colony to heat stress, is the most common way to assess bleaching response and has been measured using a lot of different techniques. The most common technique is to measure the symbiont density of a sample, and this involves counting the number of symbiont cells and the total number of coral host cells by using special equipment like a flow cytometer. And in this case, symbiont cells would fluoresce while the coral host cells wouldn't, and that's how you can separate them. Chlorophyll content is the next most used technique to measure bleaching. This method involves extracting chlorophyll from the coral tissue and measuring the optical density or fluorescence of the sample. And then measuring the color density, hue, brightness, and saturation of a sample is another technique to assess bleaching response and is digitally measured from photographs using programs like ImageJ or Photoshop. So this last technique, which is what we used, is visual bleaching scores. And this can be assigned by visually assessing the color of colonies using a 5-point scale from 1 to 5, with 1 indicating no bleaching and 5 meaning totally bleached. And these different methods of measuring bleaching phenotype require different levels of cost and effort. So for instance, measuring symbiont proportion of chlorophyll content require specialized equipment, take a lot of time, they're really expensive. Measuring the color takes a lot of time, is not expensive because programs like ImageJ are free and it's relatively easy to learn. And then the visual bleaching score is cost, time, and effort effective, but it has a lower resolution than these other methods. However, we found that visual bleaching scores are a comparable method to identifying heat-tolerant and heat-sensitive colonies. So on the left here is a plot comparing symbiont proportion, chlorophyll content, and color density on the y-axis to their respective visual bleaching scores on the x-axis. And just as a reminder, on the visual scoring scale, 1 is no bleaching, 5 is total bleaching. In this first plot, we see that symbiont proportion, which is an expensive and time-consuming method, correlated with visual bleaching scores, with each visual bleaching score bin being statistically significant from the others. Similarly, chlorophyll content, also expensive and time-consuming, correlated with visual bleaching scores, but in this case, scores were only able to distinguish between low scores, so no bleaching, moderate bleaching, and total bleaching. And finally, visual bleaching scores also correlated with the color density measurements, which is affordable but time-consuming. And again, the visual bleaching scores significantly differentiated between low scores, moderate scores, and high scores. So overall, we found that bleaching resistant and highly bleaching sensitive corals were able to be identified just as easily and effectively with the visual bleaching score system as the other more expensive and time-consuming methods of measuring bleaching phenotype. So this work aimed to address the need for establishing a simple and accessible means of identifying bleaching-resistant colonies that can be employed globally. And we showed that visual bleaching scores are a direct indicator of other common methods, though visual scoring doesn't always distinguish between small differences in bleaching response between colonies. Despite this, though, it is a reliable means of identifying individuals that are highly resistant or highly sensitive to bleaching, and this level of resolution is really all that's needed in restoration to employ a rapid and large-scale assessment of thermal tolerance. Because visual scoring is such a simple, easy-to-learn, and quick way to identify bleaching response, it's an extremely accessible method that can be employed by anyone who is interested in identifying where thermally tolerant corals might be located, given the proper permitting. And this tool can lower the barrier of entry for communities who are interested in identifying which corals on their reefs are heat tolerant that they can use in their own restoration projects. So after identifying where the heat tolerant colonies were, I wanted to understand whether that tolerance was maintained when corals were moved for use in restoration. And this led me to my next project, which focuses on the durability of thermal tolerance in Acropora hyacinthus, which is a common reef building tabletop coral. And here are some folks who helped out with that study. So a coral's heat tolerance can be shifted through mechanisms like acclimatization and adaptation. Individuals faced with varying thermal environments adjust physiologically to those environments, and that's called physiological acclimatization and is a type of phenotypic plasticity. For example, individuals living in a warm environment, shown red here, may acclimate to thrive in that environment and appear thermally tolerant, but when they're moved to a cool environment, they can then lose that tolerance and become less thermally tolerant as they acclimatize to the cooler environment. However, individuals can retain the portion of tolerance that is due to fixed genetic effects. For example, in the same warm and cool environment situation, if the organism's thermal tolerance was due to fixed genetic effects and not acclimatization, when the organism is moved to from its native warm environment to the cool environment, it's able to maintain its tolerance. So the complex interactions of these mechanisms mean that if you have a bleaching-resistant coral and you sample a portion of it for use in restoration, that coral could maintain its high bleaching resistance in its new environment, but it could also lose that resistance and instead become bleaching sensitive. To address this, we paired those acute heat stress tests that I mentioned with common garden experiments to explore the role of acclimatization on coral thermal tolerance. We collected samples from 50 corals located across 7 patch reefs in the southern lagoon of Palau, which are shown here. The yellow and red dots are sites where we collected our samples from, and the red dots are sites that where our common garden experiments were deployed. So after collection, samples were subjected to those acute heat stress tests to determine their starting bleaching resistance. Then non-heat stressed fragments from the same colonies were attached to panels for deployment at the 3 common garden sites, with 2 replicate fragments per colony deployed to each site, resulting in a total of 300 fragments in all 3 sites. Then 1 year post-deployment, we recollected the panels, and the 93 surviving fragments were subjected to a second round of heat stress test to determine their final bleaching resistance to see if the bleaching resistance pre- and post-transplantation were the same. And here's just an example of what those panels actually looked like. So the one on the left is right after a panel was deployed. The one on the right was 2 or 3 months post-deployment, just based on the growth of the algae on the panel and the growth of the corals. So in this study, bleaching resistance was quantified using both the visual bleaching scores that I've talked about and the color density, so using a photo program to measure the different qualities of color. And we found that heat-tolerant corals, or we found that 75% of colonies stayed in either the top or bottom 50% of performers after 1 year in a common garden environment. So that means we basically ranked the colonies based on their initial bleaching resistance before entering the common garden by splitting the colonies into equal quartiles from best to worst performer, with colonies that had the highest bleaching resistance spinning into rank 1 and those with the least resistance spinning into rank 4. We then performed the same ranking analysis with the surviving common garden transplant colonies based on their bleaching tolerance as determined by that second round of heat stress tests. So this figure shows the native pre-transplantation rank on the x-axis. So those in the 1 bin performed really well and did not bleach. Those in the 4 bin would have been more bleached. And then the bars here indicate the rank given to transplant colonies within those native colony ranks. So the blue shades indicate top performers and the yellow shades indicate bottom performers. So you can see that rank is generally maintained. So with native colony ranks 1 and 2 having a greater proportion of the blue top performing transplant colonies, and native colony ranks 3 and 4 having a greater proportion of yellow bottom performing colonies. The trend is particularly clear for bottom performers. So if you have a colony that has a low tolerance to bleaching, it's highly likely that it will continue to be bleaching sensitive. On the other hand, colonies with high resistance to bleaching generally stay resistant, but it's not guaranteed, as we do see about 30% of colonies dropping rank. So next we wanted to see if fragments from the same parent colony performed similarly across the different common gardens. So we made reef-to-reef plots of fragment color density for colonies that had fragments survive at 2 locations. And here what we found was that fragments from the same colony tended to perform similarly to each other regardless of where the colony was transplanted to. For example, color density data among the 18 colonies that survived at both reefs 7 and 27 showed a significant correlation, meaning they performed similarly at 2 different sites. And correlations were also positive when comparing colonies to reef 7 and 21 and 27 and 21. But the lower heat resistance at reef 21 clustered points at the lower end of the graph, so we don't see significant linear correlations between these sites. I also wanted to visualize this across all 3 sites, not just 2. So next I looked at the 14 colonies that had fragments survive at all 3 transplant sites. To remove reef effect, I calculated the mean bleaching response of each common garden site so that we could compare how individual fragments performed in relation to the other fragments at the same site. Here, blue represents a fragment that performed better than average, and the yellow kind of orange represents a fragment that performed worse than average. And colonies are on the x-axis, and transplant reef is on the y-axis, with each box representing an individual fragment's bleaching response. So for instance, colony 63 had fragments that performed better than average at 7 and 27, but worse than average at 21. And in general, replicate fragments from the same colony perform similarly across all 3 sites, either consistently performing better or consistently performing worse. However, there are 6 fragments that perform differently than their counterparts. This overall pattern is the same in this figure as the last, where we generally see colonies respond to the same regardless of location, but it doesn't always stand. Sometimes they lose or gain heat tolerance. We also noticed a really interesting pattern when looking at common garden mortality. We saw that native heat-resistant colonies tended to have a higher likelihood of survival than the native low-tolerance colonies. And this figure shows native colony rank on the x-axis and the percent survival of the transplant colonies within those ranks on the y-axis. And overall, it's pretty clear that survival decreased as bleaching resistance decreased. And this is interesting because if transplant survival and heat resistance are truly related, this would be really valuable to know in restoration efforts. It adds a whole nother reason why it'd be useful to take the time and effort to identify heat-tolerant colonies for use in restoration, because not only would heat-tolerant colonies help establish nurseries that are better prepared to survive a changing climate, but they would also increase the success and survival rates of nurseries and likely result in saved time replacing lost colonies. As you maybe heard caught in my study, we lost a lot of transplant colonies, 393, and that's actually was like a really good rate. So if it's possible that using these heat-tolerant colonies, our coral restoration efforts might become much more successful. Okay, so this chapter aimed to address the restoration barrier of determining whether heat-tolerant individuals maintain their tolerance when moved to new environments. Based on our common garden experiments and tests of heat stress, we found that corals identified as highly heat tolerant typically kept that heat resistance. However, about a quarter of colonies did lose heat resistance. And these results support the role that both acclimatization and adaptation play in coral thermal tolerance. We also found colonies with high bleaching resistance had greater rates of survival in the common gardens. And this study showed that using common gardens to identify corals with durable heat tolerance across locations is a critical step in restoring reefs with colonies that should be more resistant to future climate conditions. So after exploring the thermal tolerance of the coral, as a holobiont, so this was the coral host, the coral symbiont, the coral's microbiome, all taken into one. We didn't isolate them. We were interested in understanding more of the detail on the distribution of symbiont types in Palau and the stability of that coral symbiont partnership so that we can better understand the implications of transplanting potentially genetically distinct individuals to novel environments. So in this next project, I examined the fine-scale patterns of symbiont population genetic structure and observed symbiont response after transplantation into the common garden environments. Here are some folks who helped out with this study. So to do this, we sampled 273 Acropora hyacinthus colonies across the 39 reefs in Palau, which are shown here, colored by region. After collection, the lab generated whole genomes for each of these colonies, and given the incredible resource these genomes are and the huge data repository that's contained in them, we decided to take advantage of that and look at the symbiont genetics of these colonies. So from the genomes, we were able to pull out the chloroplast genes. We then looked at differences in these genes between colonies, as well as used those sequences to generate phylogenetic trees to distinguish between our unique symbiont types that we had in our samples. So among our cladocopium sequences, which I should say cladocopium is a type of symbiont, we distinguished two main groups of sequences, creating an upper and a lower clade, which are U and L here. Within the upper clade, we had three distinct clades, U1, U2, and U3. And in addition, we could distinguish one class of sequences that were divergent from both the upper and the lower clades, here defined as outgroup O. And those differed from about 30 sites from the in-group clades U and L, and the other clades are really distinguished by very few bases. So in the end, we have these 5 major clades in our analysis, or I'll just refer to them as symbiont types. When broken down by region, we found that symbiont type is variable. So for example, although U1, which is teal, and L, which is orange, are the most abundant types and can be found in all 7 of our regions, L is rare in the eastern reefs, but much more common and abundant in the nearby southern reefs. Other common types are even more geographically restricted. U2 in yellow is abundant in our samples, but is only found in the northern reefs. And U3 in dark pink is only found in the southern reefs. We can see these types of distributions when comparing reefs within regions as well. For example, at our southern 4 reef sites, the abundance of U3 in dark pink declines in favor of O in light pink as you move northward along the 4 reef from more protected reef sites to more exposed reefs. And a similar reef by reef distinction is seen for U2 in yellow, where U2 is only present at the reefs north of the Abyl Channel, which is just above that line there, and is absent at reefs just a few kilometers to the south. So to test the relationship between geographic and genetic distance, we just kind of to confirm what we see visually. We plotted average genetic distance against geographic distance in kilometers between our reefs, and we do see a significantly positive relationship where there is an increase of genetic distance as geographic distance increases. And while we do see these clear patterns geographically in the distribution of our clades, we also wanted to look at the genetic relationship among reefs more closely. So to do this, we performed a principal coordinate analysis, or a PCOA, using genetic distances of dominant clades. So here, points are labeled by reef and colored by dominant clade at that reef site. So if a reef had 1 L and 5 U1s, we would color it as a U1. And as you can see, we have 3 distinct clusters. So one cluster contained the 2 northernmost Oolong 4 reef sites, which house the greatest number of colonies with dominant O-type symbionts of all reefs surveyed. The second cluster consisted of 4 reefs that were dominated by L symbiont types. And the third cluster was a larger cluster of the remaining U-dominated reef sites. And even within this cluster, almost all reefs separated by dominant clade type U1, U2, or U3. Reefs within regions are also spread across the plot, and there's even plot distance between reefs hosting the same dominant clade. So for instance, the 50s in that bottom circle there, they're separated on the plot, but those were the picture on the left, those reefs that were kind of the light pink and the dark pink that I just showed previously. So they're really close to each other, kilometers apart, but they're parsing out here based on their type. And what all this means is that when we move corals for restoration, we are moving the unique genotypes of both the host and the symbiont. And while host genetics is regularly considered in restoration efforts, the symbiont genetics are often overlooked. So these results indicate that there is genetic diversity within symbionts, and it's equally important to consider this when making restoration decisions, because you can be introducing novel symbiont genes into the environment where they may not be present already. So to explore the stability of this relationship, after transplantation, we deployed another common garden experiment. So corals were collected from 23 reef sites, which are all of the yellow and red dots here, and deployed to the 5 reef sites in red. A total of 79 colonies were collected from the 23 reef sites, fragmented and placed on those common garden panels, and deployed across our 5 common garden sites. 1 year later, panels were recollected and samples were stored for transplant back to Hopkins for analysis. Using the same unique genetic sites that we use to distinguish our parent colonies, we designed primers to identify between our cladocopium clades. And we were able to successfully amplify chloroplast DNA from 91 transplanted fragments, from which we also had a native colony clade identified. Of these 91 colonies, 12 colonies changed dominant symbiont clade after 1 year in the common garden. And this may not sound very surprising, but this is the first documented instance of a change in symbiont type in an Acropora coral without a known bleaching event. So corals are known to swap out their symbiont type after they bleach, but are expected to generally hold on to their symbiont type if there's no stress event. And interestingly, 11 out of the 12 colonies that changed their clade transitioned to the clade that either dominated local colonies at that transplant reef site or to the dominant clade of a nearby colony on the transplant panel, which might suggest that colonies are sampling the symbiont composition of their environment and could slowly transition if the dominant clade of that environment changes. And this could be beneficial if the dominant clade in the environment were locally adapted to that specific environment, thus having an advantage over the transplanted colony's original symbiont type. But at the same time, almost 90% of colonies maintain their host-symbiont relationship, so it is a relatively stable partnership. So this project aimed to address the restoration barrier of understanding the implications of introducing genetically unique symbionts to novel environments. We found that there was clear genetic structure in our symbiont populations within Acropora hyacinthus. Cladocopium clades were structured between reefs and regions, with some clades only found in specific regions. Exposure to new symbiont types could shift the overall thermal tolerance of the coral, and symbiont type should be taken into account when planning reef intervention strategies. So with this restoration lens in mind, these 3 projects have provided sort of an easy method of identifying heat-resistant corals and seeing whether they maintain that tolerance and symbiont type when they're moved, which is all really valuable, but even with those questions answered, there's the issue of scaling. And Julia Rakowski, my undergrad intern, helped me with this project where I present a framework of recommendations for stewardship-based restoration as a way to equitably scale resilience-based restoration initiatives. So engaging ecosystem participants is a necessary component of gaining local support for ecosystem management and improving the outcomes of these restoration initiatives. But meaningfully involving these communities has really just started to be recognized as something that's important to do, and there's no guidance for scientists or practitioners that are not trained in this. And so to try and start to fill this need, we conducted a literature review of social ecological system and restoration literature to kind of pull out themes and factors that contribute to successful stewardship-centered restoration projects. And I'm going to go through this kind of quickly because I'll be conscientious of time, but if you have other questions, feel free to ask me afterwards. So our principles are focused on, we pulled out 6 principles: adaptive governance, equity, participatory process, knowledge sharing, consistent monitoring, and adaptability. And there are similar themes shared across these, so for the sake of time, I'll group them into principles focused on creating adaptable projects, principles focused on collaboration, principles focused on the long-term sustainability of initiatives. And I'll start with principles focused on fostering adaptable initiatives, which include adaptability and adaptive governance. Adaptability in a restoration program is necessary for the program to survive long-term. And these principles also focus on establishing a governance system that prioritizes cross-collaboration between individuals, organizations, and institutions, as well as adaptively managing projects by continuously reevaluating the success of the project and making changes as necessary. The second category of collaboration includes the equity and participatory process principles. These principles prioritize collaboration between scientists, organizations, local groups, tribal governments, and any other ecosystem participants during all phases of the restoration project, from planning to implementation. And the final category of ensuring the long-term sustainability of the initiative includes the sharing of knowledge and consistent monitoring principles. So sharing knowledge and expertise through formal and informal means of education and training allows information to be passed between researchers and the community and vice versa. And it's especially important to employ a two-eyed seeing approach, where we kind of view both the traditional ecological knowledge or indigenous knowledge and Western knowledge with 2 lenses, instead of trying to shove the traditional knowledge into a Western system. And taken together, these 6 principles can help coral researchers and restoration practitioners meaningfully center ecosystem participants in restoration efforts. Implementing these recommendations will definitely take additional time and likely money, but it's definitely worth it for the long-term sustainability and equality of restoration initiatives. Okay, so what we learned is that stewardship is an important and necessary component of ecosystem restoration and should be prioritized. Collaboration, flexibility, and respect are key themes that can be found across all 7 of our concepts. In the face of climate change, the social and cultural institutions that are holding small island communities and their worldviews together are being challenged. And while climate-induced threats such as coral bleaching and sea level rise are new challenges to these communities, small island nations have experience recovering from and adapting to other threats associated with island living. The cultural connections, knowledge, and traditions that are inherent in these communities have given them an incredible capacity for adaptation that has allowed them to thrive despite sometimes challenging conditions. And uplifting and learning from these traditional practices will be a critical component of climate adaptation strategies, including in coral restoration. So when considering future reef intervention strategies, it'll be important to take into account individual reef mosaics and their specific genetic frameworks when transplanting colonies. Using low-tech methods is an affordable and accessible means of identifying heat-tolerant corals for use in restoration, and it can help democratize these efforts. And engaging reef-dependent communities can enable restoration initiatives to be scaled globally in a just way. So some future ways that we could use this work to, well, maybe for the sake of time, I'll just skip this. But you can assess local adaptation to heat stress in important reef systems, further exploring ways to scale and intervene in these strategies, exploring links between bleaching resistance and transplantability, keeping in mind the importance of reducing greenhouse gas emissions, I will say, is still important. We can do all of this restoration, but if it just keeps getting worse, we can only do so much. And fostering equitable collaborations and employing diverse management systems will be essential employing these things across oceans will be essential for coral reefs to survive in the future. And lots of people to thank, especially the Plough International Coral Reef Center, the Columbia Lab and Crowder Lab, and NSF for funding. Okay, questions? Sorry. Any questions?
My question was, with all of the current things that you're doing to bring coral restoration, are you also looking at new stuff that is being introduced day by day? Like recently, research has come out for coral restoration based on audio. That are you planning on doing more research off of stuff that has become more current or more as of just found?
Yeah, that's a great question. So this work was all of my dissertation work, and my role right now is actually outside of this sort of coral restoration world. But I will say that the labs that I'm coming from, and maybe in the future, I would love to be able to do stuff like that. I've heard the audio recordings and things like that, and things like those new technologies I think are really interesting and neat and interesting to study on a small scale. But most of our restoration solutions or reef protection strategies, similar to the audio one, or people have talked about like dumping different types of nutrients onto reefs to see if it can help them survive. They're not really scalable and sometimes affordable. So while I think they're really valuable to learn about and see what we can do to help reefs, some of them have those scaling challenges, which is something that the coral restoration community is grappling with. But yeah, I mean, I think those sorts of things are really cool, and one day I hope to get back to the coral reef world. Thank you.
My question was about why corals can't acquire more than one symbiont and almost like build an immune system.
That is a great question, and they can. So yes, when I'm talking about these are the dominant symbiont types, so the ones that really came out, but corals are known to harbor multiple symbiont types. The cladocopium that I mentioned are known to be more bleaching sensitive than durastinium. So durastinium are another type that are more robust to stressors, but they cause corals to grow more slowly because the coral has to give them more energy to have that robustness. So they often don't like to hold on to them when they're in situations of calm. But yes, that's a great question. So that sort of shift between them definitely happens.
Any other questions? You can ask career questions, diving questions, coral reef questions.
I'm curious, how does a coral transplant work?
Sure. Yeah, so it's kind of like a skin biopsy, honestly. So like I mentioned, these corals are composed of, I won't say genetically identical because of mutations, but more or less genetically identical polyps. And so if you take, I mean, like I said, we were taking pieces about the size of my thumb. You can clip a piece off of a colony. We transplanted that, or brought them back to a dry lab site at the Palau International Coral Reef Center by boat, often wrapped in damp bubble wrap and then placed in coolers. We tried methods of kind of leaving them in bins of water on the boat, but it was just way too rocky. And they found to fare best wrapped in this bubble wrap, which I personally thought was like the strangest thing ever. Like, don't they need to be in water? But they're okay, actually. And then we bring them back and put them in these large acclimation tanks overnight before we then put them in our heat stress tanks that I had shown in that image. And we actually use garden clippers to clip them, these corals, which is kind of a funny thing.
Well, thank you. And if anyone has a question, you are more than welcome to contact Dr. Armstrong.
Yeah, please do.
So thank you very much.
Thank you.